Now we're ready for tubular reabsorption. This is the second of the processes to formation of urine. It's going to take place all the way along the renal tubules, all along the nephron, driven mostly by sodium. And then we'll look at also the different things that are being absorbed in different regions of the nephron, and then finally talk about what's called transport maximum. So again, this is the second process. We saw filtration already. This is the step where everything was moved from the blood into the tubules. And now we're going to look at reabsorption, moving things back from the tubules into the blood. So this process is going to return all the good stuff and some bad stuff too, but mostly it's going to be returning good stuff back to the blood. Now there's a couple different routes that this stuff can take. That water and solutes can move in what's called a transcellular route. The transcellular route across the cell is going to be involving moving first across the apical membrane. You can see the microvilli here, the increased surface area along that apical membrane, and then the stuff moves across the cell and then has to cross the basolateral membrane into the interstitial fluid. From there, it moves right directly into the blood. Now stuff will move from the interstitial fluid into the blood very readily because the blood has low blood pressure and it's got a high osmotic pressure because we just moved everything out. And remember, those plasma proteins are still in there, so that gives a high osmotic pressure. So that's able to pull things in very easily and sweep it away. The other route is the paracellular route. This is going to be the route between the cells, and it has to move through tight junctions. You can see here the tight junctions. Those tight junctions have little pores in it, so that allows some of the things to move through the paracellular route. In general, this slide is going to show how stuff is moving across the transcellular or paracellular route, and it starts here with the sodium-potassium pump. So sodium is pumped out, potassium is pumped in. That means we've got a very low concentration of sodium inside the cell, and so that's going to drive or generate a concentration gradient because we have tons of sodium out in the lumen because we just filtered all that sodium into the filtrate or into the lumen, and low sodium here because we've pumped the sodium out of it. So that's a nice big concentration gradient to move that stuff across. That means then we can move other things along with the sodium using that sodium gradient as the energy source to pump glucose, amino acids, some ions, and vitamins across. So this is a symporter where the sodium diffusing across changes the shape of the protein to allow us to pump the other substances across. Now, once those things are inside the cell where they'd be highly concentrated, then they'll simply diffuse into the interstitial fluid and then into the blood. Now, water has to move across the cell through what are called aquaporins. These are just little proteins in the cell that allow the water to diffuse. You can see it diffuses all the way across. And then lipid-soluble substances, since they're lipid-soluble and the cell membrane's made of lipids, they can simply diffuse across as well. The other ions, some urea, can move through the paracellular route in between those tight junctions and then diffuse into the blood. So that means a lot of our transport is dependent on those symporters, especially the important stuff, the glucose and the amino acids. And so that means we're limited, or how much we can move across, or the rate at which we can move things across is dependent on how many symporters we have. So if you look on the graph here, at low concentrations of that substrate, the substance we're moving across through the symporter, say glucose, for example, at low concentrations of plasma, we can move things across, and you can see with increasing plasma concentrations, the transport rate of that substance increases. But there comes a point in time where we reach, notice, a maximum transport rate. I can't go any more, I can't move anything else across at any higher concentrations of this substrate, say again glucose, because by this point, we've reached what's called a transport maximum. That's the limit on the number of molecules that can be transported due to limited number of symporters. In other words, by the time we hit this concentration, all the symporters are busy, and then any other substrate would actually have to wait to get a chance to be moved across through that symporter. I like to think of the analogy of toll booths on an interstate. At low, we could say, concentrations of cars, low number of cars, those cars move through those toll booths just fine. And you can see with increasing number of cars, the rate at which the cars move through the toll booths increases. But there becomes a point in time where, or I should say a concentration of cars, at which you can't move the cars through the toll booths any faster because they're all busy. And so the cars start to back up. That concentration of cars, or in this case, the concentration of glucose, that at which we reach our what's called our transport maximum, is called the renal threshold. Okay, again, renal threshold is the plasma concentration at which a substance begins to spill into the urine. In other words, any concentration higher than that, so if I have more glucose than this 4 milligrams per milliliter in this example, then I can't transport them any faster. I've reached my transport maximum. All the symporters are busy. And then instead of cars just waiting their turn, glucose can't sit there and wait its turn to go through the symporter because that filtrate is moving through the lumen. It's like a stream. And so since it's streaming through the lumen, any glucose that exceeds the renal threshold means then that that glucose is going to get swept away and become part of our urine. So when we exceed transport maximum, we end up with that substance in the urine, and we end up with a higher volume of urine because that substance acts as an osmotic, pulling water in with it because it's a solute, so it's going to make the urine hypertonic, pulling water along with it. So we end up having a large volume of urine with a lot of water in it. And that happens with glucosuria. One of the symptoms of diabetes is having glucose in your urine because we have so much glucose, hyperglycemic, we have so much glucose in the filtrate, it exceeds its renal threshold, or the transport maximum is exceeded, and so therefore not all of it can be reabsorbed, and you end up with the glucose in the urine. Now, different areas along the renal tubules reabsorb different things. You can see the proximal convoluted tubule reabsorbs quite a variety of things, and the percentages here. Notice that all of these are basically over 50%, so the proximal convoluted tubule would be voted as the most reabsorption capable part of the tubule. And then notice also that it is 100% glucose and amino acids that are reabsorbed. Also note that almost all the urea and uric acid is reabsorbed, but we're just going to get rid of that and secrete it later. In the loop of Henle, things change a little bit. In the proximal convoluted tubule, both water and solids were all absorbed in the same place. In the loop of Henle, the thing that's different is we separate out what is reabsorbed. Now, along the thin segment here, which is the descending limb and part of the ascending limb, along this descending limb or this thin segment is where you only see water being absorbed, whereas in the thick segment, which is part of the ascending limb, is where you see all of the ions being reabsorbed back into the blood. So we've just separated out where water and solutes are being reabsorbed. The reason this is a big deal is really beyond where we're going to go with this. Primarily, it has to do with these numbers here, which sets up a concentration gradient or an osmotic gradient that allows us to conserve water. And there's a section in your book on countercurrents and water concentrations or conserving water. And in that section of the book, it would describe this process, but we're not going to, or you're not going to be held responsible for that information. The distal convoluted tubule and the collecting duct, by that time, you've got 75% of the water reabsorbed. Now we're just going to do a little bit extra to it. And this is where also we're going to see where hormones are regulating the concentrations of those ions and water reabsorption. So aldosterone, which we've already been introduced when we did cardio, and ADH when we did cardio, those two are going to help with water reabsorption. Aldosterone through reabsorption of sodium. Parathyroid hormone increases reabsorption of calcium. And then the last one, atrial natriuretic peptide hormone, has the opposite effect that aldosterone does. And so here we can see an example of aldosterone. It is a steroid hormone, which means it gets to travel straight across the cell membrane and interact with a receptor inside the cell. When it interacts or binds to that receptor, it triggers other things happening, and that includes an increase in the number of sodium channels here on the apical membrane, and also sodium-potassium pumps on that basolateral membrane. That way we can move more sodium, pump more sodium out, and have more sodium diffusing in, making a bigger concentration gradient. Now that also means, though, we lose some potassium, because remember, you get sodium potassium reversed here, and we end up making a few more potassium pumps as well. That way potassium ends up part of the lumen in the filtrate, and we end up excreting more potassium. But with sodium being reabsorbed, remember, that also allows us to get more water reabsorbed, up our blood volume and up blood pressure. ADH is also involved in this. ADH, since it's not lipid soluble, has to bind to a receptor on the cell membrane. That triggers a second messenger system that they've illustrated here. And basically that protein kinase that results increases the number of aquaporins that are placed out on the apical membrane. If I have more aquaporins in here, then that means I can end up having more water diffuse across and into the blood, upping our blood volume. Now, water reabsorption then is going to be either obligatory or facultative. Obligatory is by far the predominant way water is reabsorbed back into the blood. This is what I want you to think of it as: the water is obliged to follow those solutes. So if we reabsorb sodium, for example, the water has to follow it because of osmosis, and so it's obliged to do so. In facultative, only 10% of the water is reabsorbed, and this is the part where we get to regulate how much water we reabsorb, and this is mainly through ADH. So if I increase the number of aquaporins, I can increase the amount of water that's moved across. So that ends our look at tubular reabsorption. The next video lecture will look at tubular secretion.